and on behalf of my co-investigators, I'd like to thank the Planning Committee for inviting us to be part of the press program and present to you the results of HER2CLIMB-02. These are my disclosures. Tucatinib is a highly selective HER2-directed tyrosine kinase inhibitor indicated for patients with HER2-positive, locally advanced metastatic breast cancer, previously treated with Trastuzumab and a taxane, including patients with brain metastases. The approval came from the HER2CLIMB trial, in which progression-free survival, overall survival, and progression-free survival in patients with brain metastases was significantly improved by adding Tucatinib to Trastuzumab and capecitabine. The incidence of brain metastases in HER2-positive advanced disease is quite high, reaching as high as 50%. TDM1 is a HER2-directed antibody-drug conjugate that is indicated for patients with HER2-positive advanced breast cancer after Trastuzumab and a taxane. Preclinical data and a phase 1/2 clinical trial evaluating the combination of Tucatinib and TDM1 has demonstrated promising anti-tumor activity as well as a manageable safety profile. The design of HER2CLIMB-02 is shown here. Patients with HER2-positive, locally advanced, unresectable, or metastatic breast cancer with progression after Trastuzumab and a taxane in any setting were eligible. Patients were also allowed if they had previously treated stable, progressing, or untreated brain metastases not requiring immediate local therapy. Approximately 460 patients were randomly assigned 1-to-1 to TDM1 with Tucatinib or TDM1 with placebo. The primary endpoint was progression-free survival by investigator assessment. The secondary alpha-controlled endpoints were hierarchically tested such that if the primary endpoint, progression-free survival, at the primary analysis was positive, statistically significantly positive, that would prompt testing the first of 2 interim overall survival analyses. Demographics and baseline characteristics are shown here. 463 patients were randomly assigned to the 2 treatment arms. It is notable that about 44% of patients enrolled in this study did have brain metastases, and approximately half of these patients had active brain metastases. The median number of prior lines of systemic therapy for metastatic disease was 1, and you can see that the range reaches up to 8. It is notable that the vast majority of patients were treated previously with pertuzumab, and I'd also like to point out that prior use of TDXD was excluded in this clinical trial. The addition of Tucatinib to TDM1 led to a significantly improved progression-free survival with a hazard ratio of 0.76. The median PFS with TDM1 and Tucatinib was 9.5 months, and with TDM1 with placebo, 7.4 months. Outcomes in the pre-specified subgroups were consistent with the overall population, favoring the combination of Tucatinib and TDM1. HER2CLIMB-02 is the second largest randomized study of systemic therapy following HER2CLIMB that included patients with active or progressive brain metastases. Statistical significance was not tested due to the hierarchical design of this study. However, you can see a strong trend in favor of Tucatinib in patients with brain metastases, with a median PFS of 7.8 months in the Tucatinib arm versus 5.7 months in the placebo arm, and a hazard ratio of 0.64. As mentioned previously, HER2CLIMB-02 utilized a hierarchical testing design that prompted the first interim overall survival analysis if the primary endpoint was statistically positive, which this was. With a median follow-up of 24.4 months and 53% of the pre-specified overall survival events for the final analysis. You can see the overall survival curves here. For the first 18 months, the curves are overlapping entirely, but with notable censoring beginning at month 18, these data are immature and did not meet the pre-specified crossing boundary. Another interim overall survival analysis is planned when 80% of final events have been reached. After looking at the subsequent therapies coming off study, it is notable that the vast majority of patients went on to receive subsequent therapy. Around 50% of patients in each arm received TDXD, and about 15% of patients in each arm went on to receive Tucatinib. The overall safety summary is shown here. Patients in the Tucatinib arm experienced more frequent grade 3 or greater treatment-emergent adverse events and discontinuations due to treatment-emergent adverse events. The most common treatment-emergent adverse events of at least 20% are shown here. You can see that the adverse events were consistent with those previously reported for Tucatinib and TDM1 in the phase 1 study. What is notable is the most common grade 3-4 adverse events in the Tucatinib arm included transaminase elevation, anemia, thrombocytopenia, and fatigue. And if you look at the differences between the two treatment arms for each adverse event, the most notable differences were in diarrhea, vomiting, and transaminase elevation. In conclusion, adding Tucatinib to TDM1 significantly improved progression-free survival in patients with previously treated HER2-positive advanced breast cancer, with the median PFS of 9.5 months with Tucatinib versus 7.4 months with placebo, and a hazard ratio of 0.76. The hazard ratios for subgroups were consistent with that of the overall population, and the PFS for patients with brain mets trended in favor of the Tucatinib arm. Overall survival data are immature. The safety profile is consistent with those previously reported. This is the second randomized study which included patients with brain metastases to demonstrate the Tucatinib-containing regimen delays disease progression in HER2-positive advanced disease. Thank you very much.
That was a great presentation.